Now starting from the proximal tubule. Proximal tubule comprises of two parts, one convoluted part and the other one is the straight part. The convoluted part is called the proximal convoluted tubule and the straight part is called the proximal straight tubule. Now here you can see the proximal tubule. It starts from here and this is the urinary pole, and then after pursuing a tortuous course through the renal cortex, it converts into its straight part, which crosses into the medulla to become the descending loop of the Henle. So this is the extent of this proximal tubule, and we will first concentrate on this proximal convoluted tubule. Now the length of this proximal convoluted tubule is about 14 millimeter, and its diameter is about 60 micrometer. It is the longest and tortuous part of the nephron, and it occupies most of the space of the renal cortex. Now, coming toward the histology, that what we can see under the microscope about this proximal convoluted tubule. Now this is, you can see here. Now this is one cell that is lining the this proximal convoluted tubule, and this is a simple cuboidal cell, cells. And now coming to this side, this is the luminal site of this cell, and this luminal surface of this cell, it is covered by these microvilli. And these microvilli here also covered by the glycocalyx coat, and together they form, they give rise to the brush border appearance on this aspect. Another thing here, the plasmaderma of this apical side, it also invaginates here, and it forms the apical canaliculi. Apical canaliculi are also present here, and they serve to increase the surface area for the absorption of the various molecules come across. Okay, now coming toward the basal side. You see on the basal side, the cell is resting on this basement membrane, and then you can see the invaginations of the basal plasmolemma. And these invaginations, along with these mitochondria, those are aligned. There are so many mitochondria present here, standing in a form of row, and this gives rise to the basal striations. What we can see under the light microscope here, you can see the basal striations. And then coming toward these lateral side, on this lateral side, there are invaginations and evaginations. So they are interdigiting with the neighboring cell, and so demarcation between these two cells is not very prominent on this lateral side because of these interdigitations. And one more thing, this at this end, there are junctional complexes are present here. Junctional complexes, zona occludens and zona adherens, and it's written here tight junction, but these are not the tight junctions, because the water and dissolved solute can pass between these two cells into the interstitium here. And what is inside the cytoplasm? Now you can see the centrally placed nucleus and the routine cell organelles with the abundant mitochondria. Another important thing which is present here, these are the penocytic vesicles. These penocytic vesicles, the role of these vesicles we will discuss shortly later. So Now, see, this is another aspect of the proximal convoluted tubule. Here you can see these cells are arranged side to side, and these are the brush border appearance because of the presence of the microvilli, and these are the basal striations, and outside them there lies the this interstitium of the kidney, and this is the blood capillary showing the various blood cells are present here. Now we can just to repeat and to make you remember, the proximal convoluted tubule is the longest and most tortuous part of the nephron, so occupy the major part of the renal cortex. It commences at the urinary pole and it ends by the commencement of the straight part of the proximal tubule in the renal cortex. So whole of the PCT is present in the renal cortex. It is approximately 40 millimeter long and has an outside diameter of about 60 nanometer. PCT is lined by simple cuboidal epithelium. Each lining cell contains a central spherical nucleus, usual cell organelles, with exceptionally more mitochondria. Luminal surface of the cells of the PCT exhibit a prominent brush border in histological sections because of the presence of microvilli. On the basal side of the cells of PCT exhibit basal striations. Now the cells of the PCT shows invaginations of the basal plasma lemma and mitochondria lined in rows between these invaginations, which is responsible for the basal striations under light microscope. The apical plasma lemma of the PCT shows small invaginations. These are called apical canaliculi to increase the capability of the endocytosis of the PCT cells. The cytoplasm of the PCT cells contain pinocytic vesicles to carry absorbed macromolecules to the lysosomes. Lateral border shows the evaginations and invaginations, so interdigite with those of the adjoining cells. The apical part of the adjacent PCT cells are bound to each other by junctional complexes, zona occludens and zona adherens. These junctions are not very tight, and water along with the dissolved solute can pass between the PCT cells from the tubular lumen to the renal interstitial tissue. Now coming toward the functions of the PCT. For the functions of the PCT explained by this picture, first we will have to go through this key. You see here in this corner there is a key for this figure. You can see here a circle with an arrow. It shows the active transport. You see two arrows and one circle. This is the co-transport. Simple arrow, passive diffusion, and if something is happening under the influence of ADH will be written there. Okay, now coming toward this segment. Now this is the segment of the proximal convoluted tubule, and this is the straight part of this proximal tubule up till here, where there is sudden decrease, abrupt decrease in the size of or diameter from 60 nanometer to 30 nanometer, which is the hallmark of the start of the loop of Henle. So to see what is happening, to function, the main function of the proximal convoluted tubule. The main function of the proximal convoluted tubule is the reabsorption of the water, sodium, and the nutrients. What nutrients? For example, glucose, for example, amino acids, for example, vitamins, and some other things like fractions of calcium, bicarbonate, phosphate, potassium. So these all are reabsorbed in this portion of the nephron, that is the proximal convoluted tubule. About 65% of the water and sodium which is present in the glomerular filtrate is reabsorbed in this portion. This is very important. Then comes the nutrients. You know, the glucose. For the reabsorption of the glucose, this proximal convoluted tubule is the sole site. This is the only site where the reabsorption of the glucose can take place. because once glucose crosses this proximal convoluted tubule, then it cannot be reabsorbed, and then it will be appearing in the urine. And this is what happened in diabetes mellitus, because there is so much amount of the glucose in the glomerular filtrate, and the proximal convoluted tubule is unable to reabsorb it, and that is why the surplus this glucose will pass out in the urine, and it is called the glycosuria. Okay, now what happens to the amino acids or the proteins? Now these amino acids, they can reabsorb. They can reabsorb into the interstitium from the glomerular filtrate. They pass through the cells into the interstitium, and then from the interstitium they are taken up by the peritubular capillaries. But sometimes some macromolecules are present in the glomerular filtrate, and so I have already told you that there are some glycocalyx layer present over the microvilli. These glycocalyx layer, it contains proteases, so it break down these macromolecules, and the liberated amino acids can then be reabsorbed. And even if it is not possible for the glycocalyx and their proteases, then what happens? I have already told you about these pinocytic vesicles in the cytoplasm of the cells of the proximal convoluted tubule. So these cells engulf those molecules, and then they present to the lysosome for the further breakdown. So these are the reabsorptive functions regarding water, sodium, glucose, amino acids. Okay, now coming toward the secretory function. So not only the absorption, but some secretion is also added by these cells into the glomerular filtrate. From where these secretions are coming? These are actually coming from the blood, from the peritubular capillaries into the interstitium, then into the cells, and then into the glomerular filtrate. Now, what are these? These are bile salts. These are organic acids. You see here mentioned here, the organic acids. What are these organic acids? Organic acids are oxalic acid, uric acid, citric acid, formic acid. So these are pushed into the glomerular filtrate. So in addition to these oxalate, urate, sorry for the interruption. I think some problem with the computer. Okay, I think the voice is clear and the screen is well shared. There is a doubt from the computer side. Okay, now we will continue. So I have already told you about the reabsorption of the various substances and the secretion of the various substances, including the bile salts, organic acids, oxalates, urates, and also some drugs also. They pass on to the glomerular filtrate for the rapid elimination from the body. Okay, now what is happening here? When the proximal convoluted, this is the straight part of the this proximal tubule. Now in this straight part of the tubule, it starts where the convoluted part ends, and then it ends where the upper part of the loop of the Henle starts. In the histology, it remains the same as in case of the proximal convoluted tubule, but everything is somewhat low or fewer For example, the height is somewhat low, the microvilli are fewer, the mitochondria are fewer, and then the apical canaliculi are fewer. So everything is reduced, but having all the properties of the proximal convoluted tubule. Now, again, to repeat and revise, that the chief function is the reabsorption of water, electrolyte, and organic nutrients from the glomerular filtrate. About 65% of the sodium and water filtered from the plasma in the renal corpuscles are reabsorbed through the PCT. The sodium pumps located in the basal domain of the PCT cells pumps sodium from the cytoplasm of these cells into the peritubular interstitial tissue of the kidney and increases its tonicity. Increased tonicity of the renal interstitial tissue attracts water present in the tubular lumen and forces it to pass through the paracellular route to reach the interstitial tissue. From the interstitial tissue, the water along with the dissolved solute is absorbed into the blood through the peritubular capillaries. Now, the functions are normally all the organic nutrients present in the glomerular filtrate, glucose, amino acid, vitamins, are completely reabsorbed in the PCT. Small proteins present in the glomerular filtrate are degraded by the peptidases present in the glycocalyx coat, covering the luminal plasmodium of the PCT cells, and amino acids so released are reabsorbed. Larger proteins in the glomerular filtrate are endocytosed by the PCT cells. Reabsorption of small amount of the bicarbonate, potassium, phosphate, calcium also occur in the PCT. This is about the secretory function of the PCT cells. I have already told you that the bile salts, organic acid, oxalate, urate, and many drugs from the peritubular capillaries secrete them into the glomerular filtrate for rapid elimination from the body. And this is what happened in diabetes mellitus. The amount of the glucose in glomerular filtrate greater than the absorbing capacity of the PCT. Therefore, glucose is passed in the urine, resulting in polyuria, and that is the excessive urination. Now, this proximal state tubule. Cells present in this part of the tubule contain all the structural properties of the PCT, and but less in amount. As I have already told you, that low cuboidal cell, less well-developed brush border, fewer apical canaliculi, fewer mitochondria, lateral and basal infoldings are also infrequent. The proximal state tubule abruptly narrows after passing into the renal medulla to form the descending thin segment of the loop of Henle. Now, again coming to this figure, and now we will discuss about the loop of Henle. Now, the Henle's loop or the loop of Henle. This is the loop of Henle. You see here, it is a U-shaped structure, having a thin, this descending segment, then a hairpin-like turn, then it ascends upward, and again the thin segment, which gradually, here it shows abruptly, but it is gradually passing into the this dilated portion or the thick portion of the loop of Henle, which become continuous with the straight part of the distal tubule. Okay. Now, as we know that there is a abrupt decrease in the size, where this descending, this is the straight part of the proximal convoluted tubule, having external diameter of about 60 nanometer, it reduces to about 30, from 60 micrometer to 30 micrometer, half of the diameter. And from here, the loop of Henle starts. Now, this loop of Henle, it is whole of the loop of Henle is lined by the simple squamous epithelium, except this thick part. Thick part is lined by the simple cuboidal epithelium. Otherwise, this all portion is lined by the simple squamous epithelium. Okay. Now, what is happening here? This descending portion, it is freely permeable to water. As you can see there, the water is just moving out. So 20% of the water present in the glomerular filtrate at this level, it passes out in this way. freely permeable, this descending limb. But the ascending limb and the thick ascending limb, it is virtually impermeable to water. So no water is reabsorbed from this side. All is reabsorbed from this descending limb, but not from the thin ascending and thick ascending limb. So what is happening here? Now this thick ascending limb, I have already told you that this is lined by the simple cuboidal epithelium. And this simple cuboidal epithelium, the luminal surface, it shows a small microvilli. What type of microvilli? Club-shaped microvilli are here, and there are basal striations present, mitochondria present. And so what is happening here? In this, there is reabsorption of the sodium, chloride, and potassium. So these things are going into the interstitium. You can see here by the active transport. In this region, they are going out. Okay, now as the sodium is going in this region, you know what it causes? It increases the osmolarity. And it osmolarity, it draws more water in a paracellular manner through the spaces between the cells, and the water comes out into this space, and here also in this space. Okay, now moving on to the this portion, which is the straight part of the this distal tubule. But first we should revise about the loop of Henle. Now again, repeating, a U-shaped structure having a descending and ascending limb. The juxtamedullary nephron has the longest loops and considered a typical loop of Henle. Typical loop of Henle consists of three parts: a thin descending part, a thin ascending part, a thick ascending part. After entering the renal medulla, state tubule narrows abruptly to an external diameter of approximately 30 micrometer to become the thin descending segment of the loop of Henle. After it descends into the renal medulla for a variable distance, make a hairpin loop which ascends towards the cortex as the thin ascending segment of the Henle's loop. As it reaches near the cortex, the thin ascending segment gradually increases in diameter to form the thick ascending segment of the nephron loop. This segment continues to become the straight part of the distal tubules which enters the cortex. So note these points, that which part is entering into the cortex, which part is leaving the cortex. Now the whole of the Henle's loop lined by the simple squamous epithelium except the thick ascending segment which is lined by the simple cuboidal epithelium. Regarding the functions of the loop of Henle, I have already told you that the ascending thin segment of the Henle's loop is freely permeable to water, and about 20% of the water in the glomerular filtrate reabsorbed in this segment. The ascending thin and thick segment of the nephron loop are virtually impermeable to water. Cell lining the thick ascending segment of the Henle's loop reabsorbs sodium, chloride, potassium from the urinary filtrate, and sodium ions pumped into the interstitial nephron by the sodium pumps. The accumulation of the sodium in the interstitium of the medulla creates a hypertonic environment which in turn draws more water from the descending thin limb into the medullary interstitial space, from where it is absorbed into the peritubular blood capillaries.